Yes, I think. Dejamos la puerta abierta. Ah, vale. No sé, now it's ready. Cheers. Manon, yes.
Can I press the button because I have some?
Yeah.
Okay, thank you.
Okay, so in the meantime, let's start. Okay, good morning, everyone. Welcome. Good morning. Well, the first session. So we will be starting really soon. I will just give a few information. So we will ask this. Exactly. We will ask for this row, second row to always be left empty. We have 3 presentations this morning. I will present the jury members. We have Professor Carlos Serrano from VPN, Dr. Nicolas Degri from and Dr. Perales from University of Luxembourg. For each presentation, you will have 15 minutes to present, followed by 15 minutes of questions from the jury. If there is some time left, the public is allowed to make questions on site first, and we will check also on the stream if there's any question. Remember that as the session is streamed, we'll ask you to be, obviously in respect for your colleagues as well, to be quiet, and remember that everything is on stream. So even between sessions, between presentations, please don't be loud, be respectful. Thank you very much. Okay. Manon, course will present about transposable elements in brain chemosynthetic symbiosis. You have 15 minutes when this is ready. And when there's 3 minutes left, I stand up. Then when you have 1 minute left, I'll give you a sign, okay? So you have two warnings. Make sure your phones are on silent, please.
No, it's going out of the visualization of the center. Sì. But it goes directly into the center view.
Yeah, that's okay.
No.
But not for the line. No, it should be here.
Yes. Presentation personal finance is the title.
Normally it's here, the center view.
And it shows here the
Yeah.
instead of there.
No, the problem is this
Yes.
is but not the
Because you need to duplicate this one with the line.
no, like this should not appear. It should be a normal presentation, not. Only sorry. Now. Now it should be okay.
Okay. Do we see the presentation? Do we see it? Okay. We ready to start? Okay. Sorry for the delay, Manon. You have 15 minutes.
Thank you. So good morning, my name is Manon Coste, and today I will be presenting my master thesis work on transposable elements in sulfur oxidizing bacterial endosymbionts of marine invertebrates. So I carried out this project at the Burgess Laboratory, which specializes in marine animal and bacterial physiology and biochemistry, specifically in animal and bacterial chemosymbiosis. So what is chemosymbiosis? It's a nutritional partnership between a eukaryote, which in the case of my project are all marine invertebrates, and a bacteria. So this is not to scale. The bacteria are actually much smaller. And in mussels and clams, they're found in their gills. So the eukaryote is referred to as the host and the bacteria as the symbiont. So the bacteria oxidizes reduced compounds. For sulfur oxidizers, use sulfur compounds to create simple carbon compounds that serve as nourishment for both itself and its host. These are mutually beneficial associations, and they're often long-term, and they're found in a variety of different marine environments. In shallow waters, they're found in seagrass meadows and coral reef sediments, and in deeper waters, they're found in hydrothermal vents, cold seeps, and organic falls, which I will talk about later. So the invertebrate host families who have these chemosynthetic bacteria, and that I will be discussing in my project, are Cybologonidae tube worms, Mytilidae mussels, Vesicomyidae clams from deep waters, Solimidae clams from shallow waters, and Lucinidae clams, which are unique in that they're found in both shallow and deep waters. So this is an example of an environment where these associations are found. It's a seagrass meadow, and these are Lucinid clams, two species, Lucina pennsylvanica and Kodakia orbicularis, which were sampled by a member of the Burgess Laboratory. This is another deep sea environment, so this is a hydrothermal vent. So hydrothermal vents and cold seeps are areas of the oceanic crust where there is geological seepage of compounds that can be used for chemical energy, such as methane and sulfur. And the main difference between cold seeps and hydrothermal vents is that hydrothermal vents seep liquid that is very warm. It can reach up to 450 degrees Celsius, while cold seeps have liquid that has the same temperature or similar temperatures as the surrounding seawater. On the top right here, we can see the species Calyptogena magnifica, which is a species of vesicomyid clam. And here, this is the tube worm of the species Riftia pachyptila, which sits on a bed of mussels of the genus Bathymodulus. So these are all organisms that have chemosynthetic bacteria and that are studied by the Gerges laboratory, and they study how this association can help in the adaptation to unique environments. So specifically, my project focuses on how transposable elements in bacterial symbionts can participate to this adaptation and evolution. So a transposable element is a region of DNA that can move from one place to another in the genome. It can move inside the genome of one organism, but it can also move between genomes of different organisms. There are two classes, class 1 and class 2, and class 2 is the most common in prokaryotes, and it moves with the help of an enzyme called a transposase. So the transposase is encoded by the transposable element, and it's usually the only gene encoded within this element. So these elements are very evolutionarily significant because they can lead to a lot of genome rearrangements, including deletions, duplications, and inversions. This can be nefarious to the organism as it can decrease its fitness if it disrupts important genes, but it can also offer opportunities to accelerate evolutionary processes. It can lead to exon shuffling, which can create new genes with new gene functions, and it can lead to horizontal gene transfer when a nearby gene is picked up during the transposition event, and therefore a gene can be passed on from one organism to another, and a species can rapidly acquire a new trait it didn't previously have, which can help it colonize a new environment. So this leads to my research question of how does transposase gene content vary among symbiotic bacteria, and how might this affect evolution in the context of chemosymbiosis? So to answer this research question, I developed some hypotheses. I was looking at the genome percentage of transposase genes, which is the total amount of nucleotides that are annotated as transposase genes over the total amount of nucleotides in the bacteria's genome. And the percentage of mobile elements is often used as a measure because it represents how common they are in the genome. I looked at which factors could affect this percentage in chemosynthetic marine symbionts. So I looked at genome size of the symbiont, habitat type, whether it comes from a hydrothermal vent or cold sea, shallow or deep waters, the invertebrate host family it's from, and the phylogenetic relationship between the bacteria. I also looked at lifestyle when comparing with free-living bacteria, metabolic function when comparing with non-chemosynthetic symbionts, and environment when comparing with terrestrial nutritional symbionts. So in the last three, I compared with other bacterial groups to see which factors could affect. So this is the workflow I followed to carry out my analysis. First, I gathered genomes from the National Center of Biotechnology Information, and I categorized them according to the sample characteristics and the factors I wanted to look at. I then assessed the quality of genomes that were not from the reference database by using the CheckM lineage workflow. I annotated genomes that were missing annotations with the prokaryotic genome annotation pipeline. I viewed these transposase annotations, their distributions along the genome, as well as the transposase family they were annotated as, and I calculated the genome percentage of transposase genes. Then I characterized the evolutionary relationship between the symbionts, so I created phylogenetic trees using two different kinds of genes, either the 16S RNA gene or a lot of single copy genes, and a variety of different tools. And then I performed some statistical analysis to try to draw conclusions from my results. So this is my first finding, and I found that the genome percentage of transposase genes was associated with the total genome size of the bacteria. So this was the case for both symbiotic and free-living bacteria. It was significant for both, but it was, as can be seen on the graph, more strongly associated with free-living bacteria. And so as genome size increases, there's a higher percentage of transposase genes in general in the genome. Then for my pairwise comparisons, I found that genomes from the deep waters had significantly higher percentage of transposase genes than those from shallow waters, and that bacteria that were free-living had a significantly higher percentage of transposase genes than symbiotic bacteria. As for my comparison between chemosynthetic and non-chemosynthetic bacteria, I didn't find a difference in these categories, and for my comparison between marine and terrestrial nutritional symbionts, I didn't find a difference. So I will not be discussing these, but if you have any questions, feel free to ask afterwards. So next, I compared the symbiotic bacteria between different host families. So I found that bacteria from mussel hosts had significantly higher percentage of transposase genes than all other host families, and that bacteria from vesicomyid clams had significantly lower percentage of transposase genes, with most of them having no transposase genes at all, except for one genome. So to pull this all together, I made a phylogenetic generalized least squares model. So I made phylogenetic trees, and I created a model which looks at whether the phylogenetic relationship impacts the variation in genome percentage of transposase genes. So in my model, the phylogenetic signal was quite low. So this indicates that the phylogenetic relationship between the bacteria doesn't significantly impact the variation in transposase gene percentage. And it indicates that it's possible that the factors I looked at before have a more important association than phylogenetic relationship. So in the model, I integrated the total genome size of the bacteria and the host family, and I found that these were significantly associated with the percentage. I then made the phylogenetic tree for both symbiotic and free-living bacteria, and I also found that the phylogenetic signal was relatively low, so this indicates that the other factors are likely better associated. So I integrated lifestyle, whether the bacteria is symbiotic or free-living, and the total genome size of the bacteria into the model, and I found that, as I had previously seen that there were differences between symbiotic and free-living bacteria, these differences could actually be attributed to differences in total genome size rather than lifestyle, with free-living bacteria in general having larger genomes and therefore having a higher percentage of transposase genes. So, in all, I was looking at whether there is variation in transposase gene content between different types of marine bacteria and what factors could be associated with this variation and therefore possibly lead to some hypotheses on how they are evolutionarily connected. I found that two factors that were mainly associated were the total genome size of the symbiotic bacteria and the invertebrate host family, where this bacteria is, with mussel symbionts having higher percentage of transposed genes and vesicomyid clam symbionts having in general none. So why is this interesting? This is interesting because mussel symbionts are horizontally transmitted, which means that the host picks them up from the environment. So this means that they have a free-living life stage where they interact with a lot of other microorganisms. And this can lead to more opportunities for horizontal gene transfer. And this might encourage the proliferation of transposable elements within the genome. It's also been found that these mussels have a relatively high recombination rate, so this could also encourage the transposable element proliferation. The clam symbionts, on the other hand, are vertically transmitted, which means the host passes them on from the parent to the offspring. So they have no interactions with microorganisms outside of the host. So this could reduce opportunities for a horizontal gene transfer and explain their lack of transposase genes. Also, since they have been associated with this host species for a long period of time, this could change the cost-benefit of having transposable elements, with the cost of the elements potentially disrupting important metabolic genes being more important than the potential benefit of acquiring novel traits because they're already highly specialized for their host. So these are some future perspectives or future directions in which my work could be taken. So this work could be used as a benchmark to compare with other annotation tools because I use the prokaryotic genome annotation pipeline, but a lot of different annotation tools exist, and they could have slightly different outcomes in how they identify transposase genes. So it would be interesting to see if they come up with the same amounts in the genomes. Then comparisons of the host and symbiont genomes would be useful to see if they have similar amount of transposase genes and to look at whether it seems as though transposase genes could be exchanged between the two organisms. Then I already started doing some positional analysis of the transposases along the genome, and I found that the transposase genes are clustered around the origin of replication. But it would be, with more time, this positional analysis could be done more in depth to see whether they cluster, for example, next to important metabolic genes. Indeed, complementary studies on how the transposable elements actually impact the bacterial metabolism in controlled laboratory studies would be useful to be more specific in how this could evolutionarily impact these organisms. So there are some people I would like to acknowledge for my project that have made this possible. I would like to thank my supervisors, Dr. Peter Gerges, Dr. Corinna Bruising, and Brooke Travis for their support and guidance throughout this project. All Gerges laboratory members for being very welcoming throughout the whole period and also engaging in meaningful conversations about the topic and helping me with figures. And I would like to thank the Harvard Faculty of Arts and Science Research Computing Group for their training lessons in Python and using the high-performance computing cluster. I would like to thank March Polytechnic University for the Campus World Plus Scholarship and making this exchange possible. And finally, Immersi for making this whole study and this exchange possible. Thank you for listening to
We will start now with the questions from the jury, please.
Thank you for an nice presentation.
Thank you.
This is not my subject, so I learned a lot. But some colleagues, I would like to know, these endosymbionts, are they always there? 100% of the individuals that have them, or is it variation?
So in these cases, when they live in hydrothermal vents and cold seeps, they always have symbionts because the sulfur is the main source of chemical energy, and so without these symbionts, the organisms really can't colonize the environment. So yes, it's an obligate symbiosis.
Also for those living.
In the seagrass meadow, yes, they have always found the symbionts with these animals, even though maybe they could live without, but in these cases, they have a huge advantage with the symbionts.
Do you know if anyone tried to take them away? So what's the effect if they don't have that?
In the cases of the deep sea, I don't think they have done experiments because it would be complicated, but I think they survive without symbionts, but much less well than comparative organisms with the symbionts.
And you relate to your work, the transposable elements, how important are they for the logic of the symbiosis?
So they don't necessarily need these transposable elements because there are some clams that have none at all, but it has been hypothesized that they are very important because they help exchange genes between organisms, and so this helps really accelerate evolutionary processes and helps new organisms colonize different environments, because some clams, for example, are in shallow and deep waters, and it's hypothesized that they transitioned from the shallow waters to the deeper waters, and that it's possible that these elements have played a role in helping them acquire the genes that help them metabolize other different compounds in the deeper environments.
Thank you very much.
Yes, that's really, I'm not an expert on the subject. But still, I was wondering, I'm more of a style guy.
No problem.
And so I was wondering, for instance, from what question, I guess. You said that the two relationships you found with correlation, you had was between percentages
Yes.
and genome size. You said it was significant, I was wondering whether you tested that.
Okay. Yes, I used the Spearman correlation test to test for significance, and wait, let me go back to it. And I have the Spearman rho values here. So yes, I found 0.5 for the free-living bacteria for the Spearman's rho, which is roughly equivalent to the R squared for a linear regression,
Yeah.
and 0.35 for symbiotic bacteria. So they were both seen as significant, but obviously this one was much more.
But that's a measure of
Yes.
strength of variation that necessarily of the significance of it.
Mm-hmm. Yes, though the significant is something I looked at.
Okay.
That I did the analysis on R, and so I got the Spearman's row and also the significance of the association.
Okay. Maybe be careful because there might be some assumption of normality of the microbes there.
Oh, the data is not normal,
Yeah.
but I discussed with different people and discussed that this correlation test was possible on not exactly normal data.
Okay, second one I had, it was about the, so you tested also the phylogenetic relationship
Mm-hmm.
and how that impacts your classifications for and I was wondering what was done.
Yes. So as I explained here, so I did different techniques to build the phylogenetic trees.
Yes.
I either used the 16S RNA gene, which is a gene that's often used in bacterial phylogenetics because it's relatively conserved compared to other groups, so it can help identify species. And so that I used IQ-tree for. And then I used single copy genes, which is a variety of different genes, but that are only found in one copy throughout the genome. And with that I used I used programs and pipelines for this. I used G2Tree, which is a program where you can put the genome into G2Tree and then it finds important single copy genes and creates a phylogenetic tree out of it. I also used RAXML, which is another program. They have similar methods, but I just wanted to verify that they would give similar phylogenetic trees. And then on R, I did the phylogenetic generalized least squares analysis. So you put in the phylogenetic tree. Yes, exactly. This is what it is, the abbreviation. So you insert the phylogenetic tree, and then you choose a correlation model, and it identifies whether the phylogenetic relationship impacts the percentage of transposed genes.
Okay. Do you know this one is like I'm not familiar with it. Do you know the fusion relationship are represented? Are they like for instance just in predictions or in terms of distance?
So it's a maximum likelihood model, so it gives like the maximum likelihood that these genomes are in that position. It repeats the analysis different times to make different trees and sees in which cases there are and not. And so in this case, in this model, you have to choose a evolutionary model.
Yeah.
So I used the Martin's evolutionary model, which is one that has been developed by a scientist. And this model pretty much just says how it identifies equations of how these traits should evolve if there was no phylogenetic relationship, and then it compares the null model to the model that you
So
put
yeah,
in.
you put some. I don't know if we have still time. Yeah, one thing I was a bit surprised to actually not see your specific comparison. So you mentioned at the 30th presentation the mode of transmission of the symbiotes. That seems to be quite important, and that's some specific interpretation. Okay, this one leaves outside and it's going to and so on. And I was wondering why, or if you tried to actually test this model as one factor.
So I actually wanted to do that at the beginning, test the transmission mode, although it is quite complicated because a lot of the organisms have a mixed transmission, which means they pick them up from the environment and they transmit from parent to offspring. So it's hard to categorize them according to how much they do this or this transmission method, which is why I discussed in my paper how I would like to try to find some experiments that could measure the transmission rate in a more controlled laboratory setting. But this was interesting because these plants we know are strictly vertically transmitted, parent to offspring, so they have no horizontal transmission, and these have no vertical. not much from what we know, vertical
So those are the two
transmission.
extremes.
They were the two extremes, and the others have a mix of transmission.
Yeah, I guess one thing that if you want to get even further in that for your search, you might want to do physically.
Oh, okay.
Which is a method to code when you are not necessarily super sure if this one belongs to this category or this category. You can put something in between.
Okay.
Something.
Thank you. That's interesting.
And I guess, yeah, one last one, still on that. So you made different operations on tests. So always try to explain the transverse percentage by some factors. Do you see an issue with that approach?
I do believe it is not perfect, which is why I also made the PGLS model to confirm some of the results I found here, because of course this implies that there is one factor that may affect the result, but as we know in an ecological context there are many different factors, but this was a primary study to see whether a difference was observed, which is why in the PGLS model I tried to integrate different independent different factors that could have an effect on the transposition percentage. And so the model, the PGLS...
Also in the PGLS there is not only the biology, there's also...
Yes, I put in the PGLS, I put lifestyle and the total genome size for this one, and for the other one I put total genome size and host family as variables that could potentially affect. But for these, yes, they are more simplified analysis, but I thought it could help me get an idea of which factors have an effect.
Okay, yeah. But probably didn't clear in your implementation. Yeah, because I really understood the PGLS was only.
Yes, I tried to simplify some things to make it more clear. Yeah. Thank you.
Professor Serrano? Yes, you have 5 minutes.
Just a very general question and curiosity about, I think this study are very important now from in the future, probably they will increase a lot. But is it directly for me to see which is in your opinion the evolutionary role of these results? If you started the research with a research question here on this, or it's just a matter of to explore some different data that already the team has, so you enter on top of this, or there is a clear research question on you can simplify your research.
Yes, of course. Good question. So I think, of course, this was an exploratory analysis, so from the results I was able to make a lot of hypotheses, but it's really hard to draw conclusions from this study because the aim of the study was to pull all available data together on these chemosymbioses. So I looked at all high-quality genomes that have been published, and I wanted to draw some hypotheses from them. And I also did an extensive literature review to see what was in the literature already and try to strengthen or develop new hypotheses based on this. But it's hard to be definitive on the evolutionary role because we can hypothesize how these transposable elements move, but it's difficult to find proof of their movements. But this is why this work could lead to other controlled laboratory experience where maybe transposable elements are put into these hosts and seen how.
Do you think the feeding activity, for example, the feeding strategy, is something that in your opinion can affect is something related? You analyze many.
Yes, so that was the aim for my chemosynthetic and non-chemosynthetic comparison. So I didn't talk about it because it was not statistically significant and I didn't have time. But so the chemosynthetic bacteria, they use reduced sulfur compounds, and the non-chemosynthetic used either photosynthesis or organic matter as energy. So I hypothesized that this could potentially affect whether they need transposable elements or not, and how many they need, also which types they need. But I did not find a significant difference. This does not mean that it does not affect. There could be different families of transposable elements. But since I had a large quantity of genomes, I didn't have time to go through each genome individually and look at how this might affect it. Yes. Thank you.
Thank you very much. Do we have one quick question from the public? Or online? No? So as we started a little bit late, I would suggest going to the second session. Thank you very much.
Thank you.
And now we welcome Elena Garek, who will be talking about changes in kelp abundance in southeast England. As previously, you will have 15 minutes. Now we stand up when you have 3 minutes left, okay?
Hello, Helen. So a little bit of background, brown algae from the order Laminarials and ecosystem services. For example, provides several species, including ones which are integral economically, providing reasons why kelp is a cold. And these marine heat wave events also facilitate and also events can help an image of laminar see by a very large decline. Previously, this decline has been measured sampling. As you can see, it is a pretty glamorous work. It can also be quite dangerous and tidal, slippery, and a lot of time. But more recently, there have been advancements in remote sensing techniques, which is described as the observation of a target by a device separated from it by some distance. And these techniques are particularly useful since they are non-invasive, safe, cost-effective, and more time-efficient. So, along with my supervisor, I wanted to create a way to use remote sensing data and to quantify. So I made a machine learning algorithm to allow me to quantify this sensing data, and I wanted to use the time I've seen as well. And those were my two main aims. There are many types of remote sensing data, for example, satellite imagery, aerial imagery, underwater imagery, LiDAR using light beams, and sonar using sound beams. I chose to use aerial imagery for a number of reasons. Firstly, it was really available to me from the Channel Coast Observatory, which is an England-wide project for the help of environmental management. It had a good temporal range going back to 2001, a good resolution of 0.1 meters squared. The imagery is taken at low, which is particularly useful for me since, again, kelp is found in the intertidal. They also provided true colour and false colour imagery, which turned out to be very useful. It's been successfully used in other similar remote sensing studies, so I knew it was a good option. This is the type of imagery I was looking at. As you can see, in 2001, there is quite a large amount of emergent kelp, and in 2016, it's drastically reduced. This is the sites I chose to look at. I chose them based on exploration of the Channel Coast Observatory images, looking through the years for anywhere where I could potentially see a decline, and also based on existing literature from those field surveys. I went to visit a few of the sites to ground truth and get an idea of the current abundance of kelp. Because my latest imagery was from 2022, it unfortunately wasn't possible to exactly correlate an exact GPS location to a place on the imagery, but it still gave me a good understanding. So when I downloaded the imagery from the Channel Coast Observatory, I had to piece it together in QGIS like this. And from there, I clicked to the area I wanted to look at. This helped to reduce the processing time. It would take me way too long, the computer way too long, if not. I wanted all the images to have the similar color profile to not create issues further on. And so I used a relative normalization in R to correct all the colors, like this. There were 11 images per site of 4 of false color, and the others true. And with my 9 sites, this meant processing 99 images. I was then to collect the training data from the latest imagery. I chose 5 classes of kelp, rock, green seaweed, sand, and water. I chose 50 points per class across the 9 sites, and the false color and the true color imagery, which was 4,500 training points in total. I did a quick exploratory plot to understand if the false color imagery was going to be useful to me. True color imagery is made up of red, green, and blue, and the false color imagery has the extra near infrared band. The kelp bar is the one circled, and as you can see, it's distinguished a lot better within the NIR from the green seaweed and the rock. So then I went to try my models. I used a spot vector machine model. I tried it with true color imagery, and I also tried it with false color imagery. And the false color, as I expected, performed better. I evaluated these based on the kappa statistic, whilst taking others into account. I also tried a maximum entropy model, again using the true color and the false color. And the false color once again performed better, and with the maxim performing ever so slightly better than the SVM. So this is an example. This is the type of false color imagery I was working with. These are the output, an output from the maximum entropy model, which is using binary data. So the brown is the kelp pixels that I was going to be later counting to create my trends, and the other is in purple, the other classes. And the SVM model predicts each class separately, and the brown is the kelp again. So I did this for all my sites in all my years. So these were the results from my maximum entropy model. I appreciate this is quite a lot to look at in a presentation, but overall I saw a decline from 2013 to 2016 with some signs of partial recovery, and from 2020 to 2022. And the support vector machine model also showed the same trends, which is good because they are supposed to be identifying the same thing. There are some subtle differences in particular sites, as to be expected. I also collected environmental data to help me understand these trends. I collected this from the Copernicus Marine Service, which is also freely available. I chose the closest data point to each site, and I took the sea surface temperature and the wave height. I then went on to group these by widely accepted practices, grouping a marine heat wave into 5 days or more where the sea surface temperature was above the 19th percentile of the climatological baseline. A similar method for marine cold spells, but with the sea surface temperature being below the 10th percentile. And for the storms, anywhere where the wave height was above the 95th percentile, with times merged together so that it wasn't just loads of storms if they happened too close together. So for the marine heat waves, this is every marine heat wave per year per site, with the colours referring to the intensities. The northern coast experienced more marine heat waves as opposed to the southern. You can also see a slight gap from around 2007 to 2013. I thought maybe this could be a reason as to why I had a higher abundance here, and why I saw a decline. And the marine cold spells, I also found more on the northern coast. The temperature of the water was more variable there. There is a gap after 2013, which again, maybe this is the reason why I saw such a decline from 2013 to 2016. And this is the number of storms per year per site as well. It varied a lot, as you can see, per year and per site. So it's not completely clear how these had an effect. One thing I did notice was a slight dip, low numbers in around 2011, which, once again, maybe this has something to do with the higher abundance before 2013. Something I wanted to take into consideration as well was turbidity. As you can see in this image, the water is a lot more turbid than in this image from 2016. I figured that if the water was too turbid, it might be concealing where the kelp is, which would make it seem as if it was not there when it is. So what I did to combat this, I couldn't change the imagery since it's historical. So I made a graph of the turbidity at every site. I personally rated it from 1 to 4 to see if it had an effect. But I also tried a more objective method of using the average red band value of every year. I took 5 samples and I averaged them. I tried to use it as a proxy for turbidity, but unfortunately this didn't really align with how I understood the turbidity to be, and I didn't really see any correlation between turbidity and my results, and so I don't think that it had so much of an effect. So to conclude, I saw a decline between 2013 2016 and Science of Partial Recovery from 2020 to 2022. And this is consistent with previous field studies in the area. It's clear that environmental influences do have an impact, but there isn't a single cause. And overall, it's really highlighting the value of remote sensing techniques for monitoring studies, particularly long-term ones. And for further study, it would have been great to work on more locations and more years. Because I chose to use the false color imagery, I was really limited to 2013 onwards. It would have been nice to see what happened before, and also when more imagery is released, it would have been nice to see that as well, to get a bigger picture. So a big thank you to my supervisor, Dr. Chris Jensen, and everyone that helped me along the way.
Very much. We'll start with questions from the jury. Professor Albert?
So, thank you. My first question, which species?
Across the UK, it's mostly Laminara digitata. There's also some, the scientific name slips my mind, but it's sugar kelp.
Yeah.
Those are the two main ones that are found on the English coast.
But can you distinguish between them?
That's not something that I really looked into, but they have a very similar color profile, so I think that that would be incredibly difficult. But between other types of algae, red algae, green algae, definitely possible.
But what about the other plants? That maybe it's a little bit further out in the sun.
As long as
you have
as long as they have a unique color profile, then it's definitely possible, since it's all based on the color of the pixels.
And you can distinct, well, you didn't have that kind of Fuchsteropus just above the calculus.
It's not really something that I was focusing on, so I didn't really look into that.
Because you could maybe compare with the feed studies, so you have your area model and correlate that with the feed studies.
Yeah.
So you actually can say that this is because it's a fantastic tool, but still you need to see the right species.
Mm-hmm.
Well, it's really interesting also when you correlate this to the environmental factors. But
Yeah.
what about the biological factors?
Yes.
That has been responsible for kelp disappearance on an annual basis.
Yes, this is one thing with my environmental data. It's very tricky to understand how it was actually affecting the trends I'd seen. If I had more time, I would have liked to have looked into that, and also human impacts. One thing I considered is I saw the increase, the signs of recovery I saw in 2020. We're not really sure what that's about, but maybe to do with something I thought about was COVID, like human proteins maybe.
Earlier, like experiences of kelp has been related to grazing,
Yeah,
mainly by sea urchins. But
Yeah.
there's no indication from other field data that we actually have some invasions of sea urchins in some of the years.
not that I've seen.
Okay, thank you very much.
Thank you.
Thank you. Actually, I guess we're beyond this one.
Yeah.
One remark that was really quickly. When you make these kind of comparisons, it would be really helpful to have some graphs for example too, instead of simply the time series.
Yeah.
But if you have a time series of just with the pattern of the time series, use the same time axis.
Yeah. This is the reason this is because I had the imagery from 2001 So,
Yes.
but I couldn't use it because of I chose to use false color.
Sure.
And so, yeah, I do appreciate that that makes it a little confusing. But
Yeah. And moreover, so here, for instance, you are drawing attention to an event around 2010,
around
2012.
2011, yeah. Yeah.
But your actual time series on your very start to 2013.
Yeah,
So, yeah.
yeah, I just wanted to point out that maybe that's why the abundance was higher in 2013,
Yeah.
but I can't see what happened before that without using true color in my model. So,
Yeah, so those were just looking to the markets. Yeah, I was also wondering in terms of distinction between what's scale and what's not scale.
yeah.
Do you have a way that you tried to see if you can distinguish between attached scales and detached scales? Because, I mean, sometimes you have full reaches of
Yeah.
detached scale. And
Yeah, it's a good point. I did try and look into the geology at the sites that I was working on, because some sites are more chalky, and when there's more storms, then there's going to be more detached kelp. But from my predictions, no, is the honest answer. I didn't. Yeah.
What kind of frequency do you have on those images? Or regularly as it is?
What kind of what, sorry?
Frequency for the image, the IL image you're using, how often they are?
Oh, right, okay. Around every 3 years, so I had
Okay.
like 2001, 2005, 2008. It's intermittent, really.
And you only have one task.
Yeah, just one image per year per site. Yeah.
Scales and suggestions. Okay, I have another question of the whole. Actually, it's a quite specific question. For the ground tracing of
Yes.
your machine,
Yes.
you said that you went on the fields,
Yes.
but you didn't have images that matched the times.
Yes.
So I was wondering what did that help you and what did you include that in constraining in a way if you didn't have a match?
Yeah, so what I did was, it didn't ultimately help my model, but when I visited, I geolocated where there was kelp and other classes. I made maps in QGIS so that I could just give myself an idea really as to whether the predictions were reliable. So yeah, because I wasn't there in 2022 and there wasn't an image taken when I was there, I couldn't do anything about that. But I think that could also be something nice for further study, to be able to be there for the next round of imagery. That would definitely be useful, because I saw a lot of studies that were able to do that, to use an exact coordinate and match it to an exact pixel. Yeah.
Is there any limited long time series, maybe people going regularly in the field?
Yes,
this area,
there's somebody, Ian Titley from the Natural History Museum, who helped me a lot. He has been field surveying for years.
Yeah.
One of the pictures I showed earlier, I went out with him to do some contract sampling. So that's really how I was able to tell if my results were reliable and aligning with what he had seen. So yeah, that was nice to have for sure.
Could you use that for your projects?
Well, his papers mostly graphs in decline and trends. So I didn't know if I could use use that for ground truthing exactly. But his knowledge and his images, he has like a lot of pictures. Yeah, definitely.
Everyone time. Yeah. So you mentioned that first order images perform better than triple images
Yes.
classification.
Yes.
I was wondering, is there a way to include both? Because if you just want to create a classification, the knowledge you have.
Yes. No. I could really only make a trend with one type of imagery each. I wanted to also make one with true color imagery. I didn't have time, but that's something that I would have liked to have done to see. Yeah, I could only really use one imagery at a time with the model.
Are they independent colors? The two images.
They are the same image, but with the false color has the added NIR near infrared band.
Okay, so you've got like red, green, blue in both. You've got an IR in addition in the.
Yes.
Okay, I see. So you already have all the information.
Yes.
Of the, okay.
Yeah.
Yes.
Oh, yes.
Okay, no, that's congratulations for your job, just a curiosity. So you work in an institute of zoology,
Yes.
but I wonder why they are interested in botanic study. Because I come back a little bit on the comment of there also. No? I wonder, I think it is very important to have some also idea on the biological, not only environmental parameters.
Yeah.
But also, so I wonder if the institute already has some information on animals, on basic assemblies, typical of the area. And so this is why probably they are interested in this kind of assessment as a zoologist.
Yeah. Yeah. Lots of habitat, lots of fisheries also. I mean, the Zoological Institute is a charity, and they do all sorts of research everywhere.
And do you think this is a pattern? No, because at the end you describe a pattern, but at the end the correlation and the interpretation that you try to develop cannot support any final interpretation. No? So why they increase and then there is this increase again? No?
Yeah.
Do you have an hypothesis also out of the data that you collect? There is some interpretation that you can have some new hypotheses to test, for example.
Outside of the environmental data, you mean? Yeah,
if you have time, I don't know. If you have still 2 years in front of you to enter more in deep to this topic and try to understand what is happening.
Yeah, I suppose looking into the pollution and the water quality, also the anthropogenic impacts, activity.
Because anyway, the warming is something that is happening also there. There is a general trend in the warming of the water.
Yeah, yes.
So in some places there are some already documented some adaptive processes. So when you lose, you have mortalities of species. Then the population starts to recover with specimens of colonies that have a higher thermotolerance.
Yes.
Just to put on the paper hypothesis to try to understand what you are looking at.
Yeah.
It's important to develop hypothesis and address your research to understand that. Very interesting.
Thank you.
Thank you very much. We have time for a question from the public. Anyone? Yes, please.
Yes, we also classified other biological factors like the sea.
Yes.
And we also looked at their response over time.
The what, sorry?
And we also looked at their response over
No. Not anything more than visually. The reason I chose these classes was to distinguish them from the calc. Of course, and especially because when I chose to use the Maxent model, I don't have that, but it's something I could easily, using an SGM prediction, it's something I could easily graph as well. Yes. Thank you.
And now we welcome Sari Bonnet, who will present about the effects of nutrient enrichment on
Last time I had something.
So do you mind Sorry? I don't mind. The problem is we're taking the display all channel. But then the screen blocked, they will have to restart the grid, reconnect it, and if it's that's for a reason. You can use these and just here so you can show. No, it's just really because they're just if you're ready, you will try. If it's great, it's on. You have 15 minutes. Yes, whenever you want.
Good morning, everyone. My name is Sari Konetta. I will present to you the effect of nutrient fertilization on the seasonal trace element accumulation in seagrass tissues. A little bit of a mouthful, but I called my master thesis Nutrigrass, coming from nutrients, trace elements, and seagrasses. So moving on to the introduction, seagrass are marine flowering vascular plants which colonized the oceans 140 million years ago. And they have a multitude of ecosystem services, such as the water quality enhancement and pollution filtering, among others, as you can see here. However, despite their importance, they are currently being lost worldwide. Since the 1980s, 35% of the seagrass meadows have been lost, and this is mainly due to, surprisingly, us. This is often due to nutrient inputs coming from agriculture and urban effluents and other reasons, such as propellers, as you can see on the scarring here. If we want to effectively conserve, manage, and restore these ecosystems, it's really important that we understand the effects of nutrients on seagrass meadows, especially in their physiology, for example. The species I will be talking about is Posidonia oceanica, which is a slow-growing, perennial, long-lived, endemic Mediterranean seagrass, especially known for its thick matte or rhizomes as a carbon storage in the Mediterranean. Seagrasses can absorb and remove these elements from both above-ground tissues, leaves, and below-ground tissues, roots, and rhizomes. Because Posidonia oceanica is found throughout the whole year, it's tolerant to environmental fluctuations, and its tissues often reflect environmental availability of these elements. They're often called reliable bioindicators to monitor coastal waters of shallow coastal areas. Nutrients, such the same as terrestrial plants. If we add a little bit of nutrients, moderate nitrogen and phosphorus, this usually promotes growth. But if we do too much, this often results in reduced survival due to algal blooms or ammonium toxicity, for example. Then the trace elements, iron, manganese, nickel, zinc, and copper are essential for seagrasses. The last three become toxic at high concentrations, and lead and cadmium are toxic even in small amounts. Plants, including seagrasses, have evolved complex homeostasis mechanisms to really regulate the acquisition, distribution, and protection to these elements. But currently, nobody has looked at the effect of nutrient enrichment on these trace elements in seagrasses. So that's what this project was all about. I investigated the short and long-term responses to these nutrient enrichment into seagrass compartments, being leaves and rhizomes. And I secondly looked at the metal-metal and metal-nutrient interactions. Personally, I wanted to gain more statistical knowledge. I wanted to become better at data visualization, and I wanted to learn more about seagrasses physiology. So moving on to the methodology now. I will show you the workflow that I followed, and I will highlight some of these elements here. So the first one, I was working with a data set collected by my supervisor, which meant that I had to understand the raw data first. As you can see here, this was a data set that was given to me. I had some dates, some numbers. At the time, I didn't understand, and a lot of different columns with elements here in rhizomes and leaves, a lot of other numbers here on the tables. I also had empty rows. So the first thing I did was really trying to make sense of this data set and following tidyverse and tabular data rules. I had one variable per column, which also helped me in my subsequent analysis later on. So a little bit more about the data set now. The study site was off the coast in Spain, in the Mediterranean, of course, with undisturbed conditions. The meadow had different patches, which means it was not a continuous meadow, which we also have in the Mediterranean. A little bit more about the statistical, the design. So the elements that were measured, as you can see here above, and again, in leaves and in rhizomes throughout time, on a short-term and a long-term pattern. So short-term for day. On day 0, 4, 11, 29, and 33, samples were taken of all of these elements in these compartments. Long-term, in 13 months, the old data set in 2003, going from May 2003 all the way to June 2004. 12 seagrass species were used, 6 of them were controlled, 6 of them were fertilized with nitrogen and phosphorus. They were, the fertilization was attached in vials, as you can see here. We had 9, 6 quick releaser, 3 slow releasers to have this continuous fertilization which was replaced every month. These seagrass meadows were resampled over time, which meant I had to deal with temporal autocorrelation in my modeling. That's not fun. So going through that, I had to read a lot of papers, read a lot of statistics books, talk to a lot of colleagues in person, but also online, and I came up with 3 different models that I could use to account for this temporal autocorrelation. I'm not going to go into too much detail, but I eventually went for the third one, the linear mixed effect model with an AR1 correlation structure to really capture this gradual decay in temporal autocorrelation. At one point, I was reading a book by Zura et al. And it said, figure 6 shows the type of pattern you do not want to see when you're hoping for a quick analysis. That was the pattern I was seeing, of course. So a little bit more about the statistics. So the model I used was stepwise procedures. I compared the nested models. If I had a significant non-significant interaction term that was taken out, and the reduced model was then used to test the other effects. I did pairwise comparisons with a multivariate T adjustment. And then for the multivariate analysis, I used PCAs and Spearman correlation tests. Moving on to the results and the discussion. So I could not find any consistent short-term patterns to fertilization. And often when I had significant interaction or treatment effect, this was when our sample size was reduced. Well, we know that phytoplankton can react really quickly, their elemental concentrations after fertilization. Physiologically and structurally, they're quite different from seagrasses. In contrast, terrestrial plants, which share these ancestral trails, often only have long-term assessments, which made it really difficult to really compare this with existing literature. I'm going to put detail into my graphs. A little bit overwhelming at first, but I'll explain it. So we have our control patches in gray and our fertilized in blue. And then for all of these elements, if they are in green for the leaves or in orange for the rhizome, this means I had a treatment effect or an interaction effect. For example, for zinc, on day 4, you can see that fertilized is higher than control. And the elements that are shown in gray just had a time effect. So here this is short term, so this is day. The white ones are showing no effects or no significant effects. So moving on to the long-term patterns, which I will mainly focus my discussion on now. So looking at the models, again, we're seeing very similar graph. This time season is here, so from May 2003 to June 2004. I'm going to focus on the ones on the bottom here, which all showed the same effect of an increased content of iron, cadmium, and zinc after fertilization in most seasons. So going to iron first. As I said, there's no research done on seagrasses before, so I had to look at terrestrial grasses and other counterparts to really make comparisons. And we know that in terrestrial grasses, if you add nitrogen to them, this stimulates the release of phytosterols, I hope I say it right, which actually chelates and increases the transportation of iron within these plants. There's also genes that are involved in the nitrogen metabolism that also regulates iron. Seagrasses, they're not exactly the same as terrestrial grasses. They probably have a different strategy, such as a study found that they usually increase the iron availability to root-driven acidification and the stimulation of sulfite-reducing bacteria in their rhizosphere. But these are the inferences I could only make. Moving on to zinc, we know from terrestrial plants, as I said, we have an increase, an upregulation of this iron. This also happens with zinc, and zinc often shows a higher effect, and this is also the case for seagrasses here. You can really see a significant effect of zinc, and this is the same for terrestrial plants. They share a lot of storage and metabolic pathways. Looking at phytoplankton in the lab, when nitrate was added, zinc was increased, but the authors of this paper didn't really know why. They think it's potentially linked to protein ligands. This is a PCA, so the zinc was positively correlated with nutrients represented by phosphorus, in this case only in these fertilized patches. PCAs are visualizations, so you can see they're close together, so we had a significant correlation in the Spearman's correlation test, which we didn't have in the control patches. Temperate regions, such as the Mediterranean, we know that seagrasses can incorporate nutrients, so nitrogen and phosphorus, into their tissues to use for later. So in winter, when growth is suppressed, we have a lot of nutrients, but we don't need them for the seagrasses. The seagrasses don't need them. That in warmer months, such as in the summer, we have high growth, high nutrient demand with low nutrient availability. So I propose that this is probably the same when we have storage of nitrogen and phosphorus. We might have also storage of zinc and iron in these tissues for later use, because these are elements that are necessary for seagrass growth. But I told you that cadmium also increased, right? And I also told you that cadmium was toxic. So how does that fit into the story? Actually, cadmium and zinc are very similar. So they have very similar chemical and physical properties. And we've seen that in phytoplankton, cadmium can actually substitute zinc in certain situations when zinc is limiting. And again, in terrestrial plants, we also know they have shared transporters. We also found positive correlations between zinc and cadmium in this study in the seagrasses. And cadmium was positively correlated, again, only in fertilized patches. So this increase of fertilizations increases the iron, the zinc, and as a byproduct, we are also increasing cadmium, probably unintentionally. Then, moving on to the leaves. Iron, I'm not really going to discuss so much. There's also an increase mainly in winter, but I'm going to focus on cadmium and manganese, which should reduce content after fertilization. So cadmium. As you can see here, cadmium in the fertilized patches is usually lower, but it was higher in the rhizomes. And we never see a simultaneous interaction effect that is happening at the same time. So we know that seagrasses can mobilize toxic elements from one tissue compartment to another. So we hypothesize that this is to protect the photosynthetic function under fertilization. Cadmium is mainly toxic for photosynthetic functions of seagrasses, so it makes sense that it would move this away from these important photosynthetic elements. We also know in terrestrial plants, if you add phosphorus, this often lowers cadmium without affecting the root uptake. There's not that much known about it. The last one, manganese, as I said, we have lower content after fertilization. And we know in terrestrial plants, if we add nitrogen and ammonium, this reduces the expression of manganese transporter genes. And ammonium also competes with manganese 2+ for uptake sites. On top of that, we also have metal-metal interactions. So we know that iron and manganese have antagonistic relationships where they compete for transport proteins, and they were also only negatively correlated, so showing this antagonistic relationship in fertilized patches. To conclude, so as I said, the rhizomes, we had an increase in iron, cadmium, and zinc, while in the leaves, iron was increased in the winter, cadmium and manganese were reduced because of this strategic below-ground storage that we proposed, and maybe to protect these photosynthetic tissues from toxic cadmium. We also have these metal-metal competitions going on. Think about zinc and cadmium, iron and manganese, which often shore these uptake and transporter uptake pathways. As a conclusion as well, I told you about this bioindicator and biomonitoring role of Paleoceanica. Under fertilization, long term, this might clearly change. So for monitoring, we should really try to do this short term when we're comparing against sites, for example, or within the same season. Seagrasses are also an entry point to trace elements into food webs, so this might alter trophic transfer dynamics. And future research should try to unravel this molecular and transcriptomic pathways involved in this uptake, sequestration, translocation under varying conditions, especially now that we are still putting nutrients into our coastal shallow areas. My personal conclusions for this master thesis, did I gain more statistical knowledge? For sure. But the more I know, the more I realize I actually don't. I do have better data visualization skills, and physiology, very interesting, very important, but it's not exactly what I want to do. And this is also not what I will focus on for my PhD, because I will focus on seagrass restoration and ecology. Thank you so much.
We'll start with the questions from the jury. Professor Arbeit, please, if you want.
Thank you for a really nice presentation, and really good that you can use the old experiment again instead of reuse it. And my question is a little bit about that. Do you know what the main focus could have been as these elements? Because then they would have analyzed it before. So, what was the main thing with the experiment, and can that in some way be correlated to your results?
So the original experiment was done during the PhD of my supervisor, but she never got to analyze the data. I was going to go back to the methodology. So originally, she did a cross-sampling design where 3 of them also had organic matter added to them, but that didn't work because of the very big grain size. So yeah, we have this fertilization and control and eventually to work with, but it was part of a monitoring study. So they wanted to look at bioindicator species of seagrasses in the Mediterranean. Is it working, yes or no? And does fertilization affect this? But she never had the time to really dive.
But in the data set, you didn't have like the number of grazers or
No. And I tried to find more. I didn't have temperature. I didn't have a lot of environmental variables to work with, which I really would have loved to have. And I tried now in 2025 to go back to the data and looking at the Copernicus data sets, for example, of looking at satellite imagery, but I could not find specific environmental drivers of this. Yeah.
Because I know that they did similar experiments all over Europe, well, all over the world, maybe with fertilization. But then they looked more at grazers and fish.
Yeah. The colleagues of my supervisor were also exactly looking at grazers and interactions with that, but I did not have any of that data for this data sets, sadly.
No relation to what they found. Still, to couple this with other effects that they actually saw when they did these
Well, the fertilizations, I think they definitely found more epiphytes, for example, on the leaves growing, because not only seagrasses are going to take up these nutrients, it's also the epiphytes growing on top of the seagrass blades, and this also attracts a lot of herbivores, such as fish, to then go into further of the food chain. But no,
But in relation to your results, the iron will that be competition with the other algae for this, or what do you think?
But what we know in terrestrial plants, if you add this nitrogen, it upregulates this iron. Iron gets taken up more. It gets released easier into the seagrasses. So we have more iron in there. Do herbivores prefer that? Potentially, it's a limiting element also for humans. That's why there's a lot of research in terrestrial research. What they're trying to do with wheat and rice plants now is adding fertilizers, because humans have an iron deficiency, but what they're also seeing is exactly what I'm seeing. Zinc also goes up, but cadmium also does. So genetically, they're trying to find, can we increase iron by adding nitrogen, but can we stop zinc and cadmium following through these pathways? But I'm sure it has an effect, yeah.
Thank you.
Yes, I guess I'm maybe going to start with a remark, and that's not to say it was a great presentation. I thought it was a bit fast, even for me. There was a lot of things in there. Obviously, there's a lot of work, nice work to present. But yeah, we try to make more shooting is out, maybe try to make more shows off. Sometimes we play with this, but that doesn't mean it's bad. The slides were really clear, so it was easy to follow, but a bit challenging. Okay, so I guess continuing on that remark on this pattern of increasing iron and zinc in connection with the utilization. And you emphasize that it's so yeah, basically helps when you have more nutrients, you just have metals from the. I was wondering, did you try that in terms of stoichiometry?
Yeah, I didn't look into that, no.
Do you think it would be interesting? I'm not super familiar with plant physiology generally,
Yeah,
but I
so
know that's okay.
it would have been really interesting to also look at carbon, nitrogen, phosphorus relationships and how that's changing. But in the long-term study, 2 seasons were lost. So that really reduced that. That's why I'm also using phosphorus as my proxy for nutrients, because it was highly correlated. It has showed high collineality with nitrogen, so I use that as a proxy to represent the other elements. But yeah, I think more data would be necessary to really look at it.
I had another one, again, the statistical line and a more of a multivariate statistical line. So that's going to be the PCAs.
Okay.
I was wondering, so you've made 2 of them?
No, I made like
Like,
16
I mean,
of them
for
eventually.
instance, for roots, for rhizomes, you're presenting 2. Right? So that's 2 different PCAs, right?
These are 2 different PCAs. We have it in fertilized and we have it in control patches.
Yes. And I was wondering, why did you choose to separate and control?
I'll show you. So going to my supplementary material. Okay, so we know that rhizomes and leaves have very different elemental concentrations. This is shown by this PCA, very clear driver of this PC1 is rhizomes, leaves, very different elements. That's also why the whole analysis was actually split up, because we know from previous literature there is a difference between that. And then I wanted to look, is this relationship changing because we're adding this fertilizer? So that's also why with the other PCAs I split it up like that.
I mean, does that explain why you have like different PCA for leaves and reasons? But for controlled fertilizers.
Because I wanted to know if this relationship was changing in fertilized. So if I add nutrients in a sudden, is this relationship changing between these elements or not?
Between the different variables?
Yes, okay. Variables being the metal elements. Because if I do my correlations, if I look at all my correlations between leaves and rhizomes, I also split it up. For me, it didn't make sense to look at correlation elements in control and fertilized if the whole experiment is about the difference between adding fertilized or comparing it. I wanted it split up. And the PCS is also for visualization. Because when I added them together, these relationships, if I then put the Spira and correlation tests on them, it didn't visualize it properly when I combined them.
Yeah, you put the first structure that's super important together.
Say again?
There's on the leaves at least, temperate structure is clearly more important than the first.
Yes, exactly. And that's a pattern I found throughout most of it. Because only some species.
Well, I mean, it's very clear from the bias, but I mean, if I want to scratch a little more the material analysis, you get an idea of where you could control on initial activities.
How we could control for what?
For like time and
no. That's why the main focus of my thesis was on the univariate analysis and incorporating this time and temporal autocorrelation. Because if you're going to have more iron on in the beginning, you're also going to have more iron later on into the study. So that's really with multivariate analysis. I don't know if you can tackle it, but I'm sure you can. But I mainly focused 2-3 months on finding the right model for my univariate analysis. And PCA is more for the visualization and then the correlation tests later on. Thank you.
Okay, now, from my side, just curious, not my field, as you know, but I would like to better understand in which way you deploy this, so the system of deployment. Because just to better understand the biological availability of these elements for the seagrass and the different level of exposure to this treatment of leaves and so on. So just to better understand, so there is a bias there. So you put the fertilizer and these are slowly released.
Yeah. So 22 years ago, they went in these seagrass patches and added these vials, which some had slow releaser, some had this quick releaser. And every month they replaced these vials to have a continuous enrichment throughout the whole 13-month experiment. Of course, we double-checked. More nitrogen and more phosphorus was actually being incorporated both into the leaves and into the rhizomes. So it was being taken up. But yeah, it is added in the water column, and it is assumed that it will eventually go down and get taken up by both elements. This is the whole design of the work.
Where you do the experiment again, you change something, or you think this is a good way to test this?
Well, first of all, I would have probably tested it in the lab, because we have never done anything like this with seagrasses. And adding all of these environmental variables immediately into this, I think there's a lot of confounding factors that can play with this. And from the studies I've been reading, you actually need benthic chambers, and you have to put them in there and then add these nutrients, not just like this, where they can just spread out to all other patches, basically. The patches were separated quite far from each other, and preliminary studies were done to check for this. But I would, if funds were not limited, I would use benthic chambers to control for this, and then look at rhizomes, leaves separate, add different nutrient elements, and I would also have nitrogen and phosphorus separately and not together. Because now, what I really struggled with is in terrestrial plants, they only add nitrogen, and they explain the effect on iron or zinc. I couldn't differentiate between that. I had fertilizers being added both at the same time. And on top of that, the fertilizers used here is the osmocot that is also used in agriculture. We know that with varying temperature in seawater, this is also variable. So there's a lot of things that couldn't be taken into account, but being said, we did see that nitrogen and phosphorus was being a lot higher in these fertilized patches in both compartments.
The risk to do is a lot of work, there's a lot of leak of energy, you know, to at the end arrive result that you are not sure at the end if they are actually not true or but anyway, for sure this is an interesting approach, and considering your interest now in restoration. Do you think this kind of results can be a support in some way, the effectiveness, or can change the result or ameliorate the result of restoration, or it is something more related to a risk of pollution, so something to avoid? In which way do these results can be supportive to the next step, for example, your restoration?
Yeah, even though I don't want to focus on physiology in my future career, I did take a lot of these ideas in there. So we know that if we add fertilization to these seagrasses, they often grow better. If we don't add too much of it, doesn't become excessive. But of course, we would have to do studies on what is the growth rate. Are they growing better? Are they growing faster? But for example, I think in Sweden, there were studies done where they transplanted seagrass with metal spikes with iron in them. And they found iron as a micronutrient for seagrasses that were actually growing better. So I am playing around with the idea of using, especially in mesocosms, to play around with these nutrient and elemental concentrations. How can we have the starting phases of seagrass restoration? Think about seeds or fragments. How can I enhance that, produce them quicker, bigger, better, and then transplant them out? I don't know if I would add fertilization into the environment, because I think for my PhD, for example, I want to try to not use plastic and not use adding these kind of elements into the environment. And I know a lot of vials were also lost during this study. So
Anyway, considering all the experience that now we have, in fact, you probably compare also and try to understand comparing the terrestrial system. No? Now we have a lot of knowledge about the error that we did with fertilizer, adding elements in soil, adding soil. So, I hope that the errors that we did on land will not be applied.
That's why I would keep it to the mesocosms, controlled environments, to not pollute the environment even more and change and alter not only seagrasses but all the other organisms living in there.
In general, when we alter natural equilibrium.
Yeah, but we're destroying them.
Sooner or later, we realize it.
Yeah.
Okay, good.
But to enhance, to a bit quicker, it's possible, I think.
Thank you very much. We have a question from the yes.
As you know, I think seagrasses can form massive flows of kilograms I was wondering the distance between these patches. What's this? Between those 12 patches?
They were at least 10 meters apart.
10 meters apart. Is it possible that you do it on one individual? Imagine it's one flow. It's one genetic individual. Is that possible? The results we see are just experimental on one individual. Yes.
think then this can generalize this data? Or what should we do then to improve or be about these elements?
Yeah, if there are all the same individuals and I have a lot of within the same, do a lot more experiments in different places with different conditions. And of course, genetic testing, that if money is there for that. We use these patches as replicates because we were like, okay, they are distinct units, but of course they could all be a clonal species. They are.
We had another question, yes, please.
One is a curiosity, just since this work was done more than 20 years ago, how is the status now of, if you have an idea of that area regarding the molecules? And second, maybe I lost the presentation, but have you checked if there were any differences in the quickly fertilizer and the slowly fertilizer or the same? So that you notice anything on the effect on the
So the corals, they were added like this. So 9 within each pet, we had 9. 3 of them were slow releaser. 6, just to have this continuous fertilization. And the second question,
if you had any information on now in the needles, now in the area.
What I found out by using this older data set is like back in the early 2000s and late 90s, there was a lot of research on nutrients. And we kind of stepped away from this, like 22 years later, and I still cannot find any study anywhere in the world, which is very frustrating, that has looked at this even in the lab. And in recent years, I think we're coming back a little bit. I found a few studies that are doing these benthic chambers, as I explained before, but currently there's not much known there, especially not related to fertilization. The seagrass patches are still there. There's being research done. The patches are still there. If they're expanding or decreasing, I didn't read anything about that. But it's near Blanes, which is a big research center, so there's a lot of research being done there.
Thank you very much. I think it's time to close the session. So.
Do you want me to leave it? It's so much nicer to present. I walk a lot. Yeah, so you did. Yeah, so it was really nice. Thank you. So I was always
session. In this session, we have 3 speakers, same rules as earlier, 15 minutes for the presentation, 15 minutes for questions. At 2 minutes, I'll try to wave at you. You may or may not see me, and if you're on the time or just a little bit over it, I'll stand up, so that's an indication that if you haven't finished already, you need to wrap up. Thanks to our 3 jury members. So we have Laura, Martina, and Chiara. Thank you. So the 3 jury members in the front. Thank you to our audience, and most important, thank you to our speakers. So up first we have Sarah, who's going to present your master's thesis research. The floor is yours. Good luck.
Thank you. Okay, good morning. My name is Sarah Sanders, and I'll be presenting you my thesis titled Bridging Innovation and Restoration, a systematic review of artificial reefs and case study for Mediterranean conservation. So to begin, as I'm sure we're all aware, biodiversity loss is accelerating in marine ecosystems, but recently artificial reefs have emerged as tools for restoration and conservation. The United Nations defines artificial reefs as submerged structures that are placed on the seabed to emulate some form or function of a natural reef. And while they were originally intended to enhance fisheries, modern ARs now serve many different purposes, from biodiversity support through habitat creation, ecosystem recovery facilitation, protection of sensitive habitats from destructive activities, mitigation of localized disturbances, and even creating opportunities for research, education, and recreation. So within the context of the review and the need for nature-inspired designs, these are the aims of the review. First is to characterize the evolution of AR design over time. 2, to contextualize a nature-inspired AR for deployment in the Mediterranean. And 3, to develop evidence-based recommendations. And for the purpose of the review, we defined ARs both by purposefully deployed structures and opportunistically deployed structures that were subsequently studied for their reef effects. So now moving on to materials and methods. The search resulted in 1,854 records, and we followed the PRISMA 2020 guidelines for a standardized review. And after screening through the papers, it resulted in 326 studies being included in the review, spanning from 1975 to 2025, so 50 years. And this is the data that was extracted from each of the papers. So on the left is just some basic identifying information, and on the right is data that was specifically included for the analysis and specifically regarding the artificial reefs themselves. So within this context and for nature-inspired designs, BORA is the case study I'll be talking about. It's a nature-inspired design. It's a newly patented AR that was designed to create hard substrate for the recovery of coralligenous ecosystems and their associated fauna. It's made up of cubic frames, which are each 2 meters long, the connections between the frames, and then shelves of varying lengths between 2 to 4 meters. And the frames and the shelves are both made up of concrete with stainless steel reinforcement and then covered with a mineral or organic material to help increase settlement of marine organisms. And the shelves can also be tilted at various angles. This is to create the typical terraces and overhangs that you see in a coralligenous community that you can see from this image right here. So in order to have a successful deployment, this is where the deployment will be. This is at Dishishao on Gallinara Island in the Ligurian Sea. So the island is a small island. It's located about 1.5 kilometers from the coast of Italy. It's uninhabited, but there are so many activities that go on there, commercial and artisanal fishing, as well as anchoring, especially during the summer seasons. It's been identified as an area of important habitats and of high ecological and biological interest. It's a site of community importance under the Habitats Directive, a special area of conservation, and a national regional reserve. And for the deployment, 6 BORA structures will be placed on a detritic seabed at 39 to 40 meters depth, occupying a total area of 100 square meters. 3 of them will be used for restoration purposes only, and 3 for restoration and dive training. And to have a successful deployment, these are the hydrodynamic models and simulations that were run considering a 1 to 20 scale prototype. it had the two parallel shelves that are equidistant from each other, tilted at 20 degrees. And this was based on the real-world conditions of Gallinara Island. So based on the max local hydrodynamics of a current flow of 2.5 meter per second and a depth of 40 meters. Then based on these models and considering the species naturally present in the area, we then created a schematic drawing to inform the organism placement that will be deployed. So now moving on to results and discussion. Regarding the results from the systematic review, we can see for temporal trends, research is expanding, reflecting the general growing acceptance of ARs being used as tools for restoration and conservation. We see a shift around 2005 towards ARs with an objective focused on ecosystem enhancement, which reflects the growing movement towards ecosystem-based management in marine systems. In 2020, we see a shift of an increase of research and method testing focused ARs, which aligns with more innovative designs. And there are some other minor objectives that we see in the literature, but there are still important things to note. So for instance, recreation-focused ARs focus on improving surfing, increasing tourism, and even creating diving opportunities, which reflects the multifaceted benefits that ARs can have for coastal communities. Regarding the geographic distributions of ARs, these are the 6 marine ecoregions with the most publications regarding ARs. So the top 3, as we can see, is consistent with robust policy frameworks in Europe, as well as the general predominance of publications being in the Western and Northern hemispheres is consistent with greater funding availability, longer established marine management programs, and greater research infrastructure. But in general, ARs were deployed globally, and ecosystem enhancement emerged as a primary objective for most of them. Secondary objectives, however, did vary based on environmental and socioeconomic contexts. So for instance, we can see in the Mediterranean Sea that research and method testing, recreational opportunities, and coastal protection were objectives that were notable, which reflects the high coastal populations, intense tourism, and the need to balance economic and ecological interests in this area. Furthermore, in the Atlantic regions in the Sunda Shelf, we can see coastal protection and fisheries management as notable objectives, which also aligns with these varying areas where sea level rise, tourism, and commercial interests are pressing issues for the future. So, as we can see these regional variations, it's important to tailor ARs to their local socioeconomic and environmental context, and it reflects the ability of ARs to incorporate multiple functions in order to maximize and optimize their benefits. Regarding the habitat distributions of the studies, ARs were most studied in a sandy and soft sediment habitat context, likely for its accessibility, and possibly related to some objectives like coastal protection. This is followed by reef and reef-adjacent habitats, estuaries, and coastal wetlands. Despite being ecologically important, seagrass and mangrove habitats were rarely studied in an AR context, perhaps due to their sensitivities to disturbances, as well as this just may not being a great option for restoring these habitats. When we focus, just looking from the left chart to the right chart, so focusing on just publications with an ecosystem enhancement objective, we can see that sandy and soft sediment habitats decrease a little bit, while reef, estuaries, coastal wetlands, and even seagrass and mangrove habitats do increase, suggesting these are becoming greater focuses for restoration-based approaches. So now moving on to material and structural trends. Prior to 2000, material and shape were more evenly distributed across categories. However, after 2000, we see it become largely dominated by concrete and geometric shape, which likely reflects concrete and geometric shapes' ease of replicability, ease of manufacture and deployment, cost efficiency, things like that. However, we do see that in recent years, experimental materials and structures that mimic natural reef colonies and floating and suspended structures are growing, indicating a trend towards more structurally complex and innovative designs. Furthering on with material and structure, Concrete largely dominates across different objectives related to ARs. For structure, basic geometric shapes are the most frequently implemented, as I said, but this is closely followed by modular and stacked structures. But in general, we can see the design differs across functional goals. So for instance, ARs with an objective towards coastal protection largely incorporate large-scale industrial designs made out of concrete and rock, as this is most effective at dissipating wave energy and is easier to manufacture and deploy on a large industrial scale. ARs focus on fisheries enhancement and management, often incorporate modular and stacked structures of dome and pyramid shapes, because these can be tailored for a specific target species. So overall, AR design is a critical determinant of ecological performance and should be tailored to the local environment. And for material and structure, incorporating these specific trends. So for structure, incorporating voids and crevices, or material incorporating a certain biomimicking material, this is representing an important trend that we're seeing in AR design and creating nature-inspired designs. So within this context of nature-inspired designs, we have Bora. This is the results from the hydrodynamic models. You can see the current velocity taken across the 5 different planes. So each of these planes is a snapshot that we see from them. We see, in general, the current velocity increases between the 2 shelves and decreases on the top and the bottom of the structure. And this is important to understand just the general design of Bora and the context that it will be placed in, and then allows us to better achieve Bora's goals. And so using the results of these hydrodynamic models, we were able to develop this scheme of organism placement. And so in reality, not all of these organisms would be placed on one structure, but this just gives an idea of where certain organisms could be placed. So these are all key target species that are found at Gallinara Island. And so the ones that you can see are photophilic and prefer low hydrodynamism are placed on the top of the structure where the current flow is low. The ones that prefer high hydrodynamism are placed in between the shelves and on the terraces where there's high hydrodynamism and high current flow. And ones that can support greater sedimentation are placed on the bottom and the edges of the structure where sediment may build up. So finally, regarding the monitoring practices that were found in the systematic review, most studies occurred in the first 10 meters of the water column, and over 60% did not mention monitoring at all, and there was in general a lack of long-term monitoring across depth ranges. When there was monitoring incorporated, short-term efforts were reported twice as much as long and mid-length efforts combined. And so short-term efforts may capture some initial colonization or some early changes, but in general, it misses long-term ecological processes, key successional changes, and can miss unforeseen consequences. And so it's important to incorporate long-term monitoring, as this helps us understand whether an AR actually achieves its intended objectives or inadvertently contributes to negative consequences. In conclusion, this systematic review characterized trends of AR design, which will help improve the restoration-based approaches of ARs in the future. It contextualized nature-inspired designs, and in particular, nature-inspired designs like Bora offer a promising solution for ecosystem restoration. And we do intend to deploy Bora this year and subsequently monitor its effects. And even still, with the growing ecological goals of many AR projects, there are still persistent gaps that remain regarding their long-term monitoring and evaluation. And so it's important to prioritize consistent and extensive monitoring frameworks moving forward. So in order to advance AR research and AR science, it's important to promote this interdisciplinary collaboration in order to move ARs past just the mitigation phase towards truly restorative outcomes. And I want to say thank you to my wonderful supervisors, IMBRC, the hosting university, and my friends and family for their support. Thank you.
audience if you have any queries or comments, and if we have any online. So, three members of that.
Okay, Laura. Okay, so thank you very much for the presentation. I enjoyed a lot with a lot of information. And I have to be honest, I'm not an expert in crowd restoration, but I have several questions, most of them are curiosities. And the first thing that comes to my mind after seeing all the representation is like, why this in long-term monitoring is not so important on the reviewer or on the reader like the because there are papers for a long, long time ago, but they don't continue with the monitoring in the scenario of achievement and everything.
Yeah, I think in general, from what I found, is a lot of times it's a lack of funding availability when starting from the outset of a project. You usually like create the project and then long-term monitoring, it needs to be at least 10 years, and so it's hard from the initial conception of the project to plan to do that for 10 years following. And so I think a lot of times it's that. And just, yeah, I think a lot of times it's a funding issue. It's also an issue of different stakeholders being involved, because like maybe the person that's employed it is a researcher, but it's in some communities C, and so you have to incorporate multiple stakeholders. You have to incorporate the local community, local fishers, divers, whatever. So it's a complex, tricky issue, incorporating lots of different people, and I think that makes it complicated to plan to do that for that long and set up a thing for that long. So, yeah.
Yeah, I'm completely right. And I'm curious, why would the Bora device exactly at 79 or 50 meters and not, for example, at different depths and have replicates of different depths? Why do you choose that particular community?
Well, coral indigenous communities thrive at a range of depths, but really at 40 meters to around 120 meters. So that's initially why that depth of 40 meters. And then that location is just, that's just, I'm not exactly sure. I'm not fully involved in the deployment, to be honest. I'm sorry.
Okay, no,
I'm not fully involved with
I will change
deployment.
my question. Do you think it would be interesting to have replicates at different depths?
I definitely think it would be. This is just the initial deployment, so that could be something that is done for the future to see differences at different depths. Yeah.
Okay, thank you very much.
Yeah, thank you.
I also have a question, also curiosity, about the graphs resulting from your ranking. And I was a little bit surprised that in the Mediterranean Sea, there's really a few percentage of papers talking about fishery management, which is surprising because the Mediterranean Sea is really fish sea. So what did you find in your revenue about this particular topic?
Yeah. In the Mediterranean Sea, most of them were focused on enhancing the ecosystem, which can incorporate fisheries as well. It was just that this was maybe their primary objective and what they stated in their paper. So it could be that they hoped that that was maybe a secondary kind of additional benefit from their deployment, but maybe not their primary goal. Because yes, it would make sense for there to be more fisheries management in the Mediterranean Sea, but these are the primary goals, so it could be that it's a secondary objective, because as I talked about, ARs often provide many different benefits, so even if they're deploying it specifically to restore an ecosystem or provide coastal protection, it's likely that it could potentially enhance fisheries as well, so it could be an added benefit for them.
This is general all
Yes, the left one is, and then the right one is specifically with publications that have an ecosystem enhancement objective.
Yes, I reserved apart from this apart from Bora, did you find any other AR that is completely native-based?
Yeah, there are quite a few labs. It's definitely a new thing, but I would say especially in the past 5 to 8 years, there are a lot of people doing nature-inspired ARs. There's a big one. They actually had a presentation in the last session on it. There's a lab called Reef Design Lab that's based in Australia. They do lots of different ones that are fully inspired by reefs in different types of reef as well. So like, yeah, different, but depending on the ecosystem that they're placed in, they have lots of different nature-inspired solutions. And so there are lots of different ones from mimicking coral reefs to mimicking oyster reefs to even creating like little rock pool type situations. So there's definitely in recent years there have been a lot of new ones, yeah.
Any questions from the audience? You're off the hook. Well done, Sarah. Very much.
Where are the fights?
Okay. Stop with the streaming, so it was chill for a minute.
Is it problem for you?
Like, it's difficult to hear
Over there, it's noise, there's a lot of noise.
Yeah.
Oh, yeah.
So,
Is it a microphone for?
Is it like a?
Oh, you
Is it
have
hand? Okay, so I will be holding two things. Perfect.
You've got the power.
Speaker. No speaker. Sure.
You can't see video.
And actually, there's no hard checking that technically that you can hear it now. Can you hear?
Is it okay?
yeah.
Is it good online? Is this good online? Can you hear it online?
No, that's that doesn't hear it online.
Yes, I was planning to project, but I guess I have to move.
Okay, that's is it custom? Yeah, I will be okay now.
This is a good delay. This is a good delay.
Catching up.
Maybe now you're testing. There is.
You can hear testing.
So 2 minutes late.
It's okay. Yeah.
Not too low because maybe
Too low or too loud?
Like this is okay.
Great.
Okay, so you're welcome to my conference, I guess. Is this working?
Okay, so I'd like to introduce Noor, who's next up to present their master's thesis research. So over to you, best of luck.
Thank you. Okay, welcome to my conference. Thank you so much for coming to my Thesis Effects. I'm Noor Bouquet, and today I will present you comparing survey techniques to monitor collagenous habitat in the Mediterranean Sea. Work that was done here at the University of Danimarca in the Zoology Lab under the supervision of Dr. Mentas and Dr. Bonetta. So first, a little context. As we know, the increased climate pressures such as heat waves and acidification in the Mediterranean combined with anthropogenic pressures such as encountering and fishing have led to a general loss of biodiversity in marine habitats. To prevent this, okay, I'm sorry, there's a format problem on my PowerPoint. Never mind. To prevent this, there has been implementation on several international frameworks, such as the EU Marine Strategy Framework Directive and the new EU Nature Restoration Law under the Biodiversity Strategy for 2030. There was also regional implementation of an action plan under the Barcelona Convention, the UNEP-MAP action plan specifically for collagenous habitats in the Mediterranean. And I just wanted to show you the marvelous collagenous habitats and the beauty and importance of it. So here you can see two pictures of the characteristic habitats. And basically it's defined as biogenic reef that is composed of mostly three-dimensional structures, such as here Gorgonians and calcareous algae, and you have erected sponges, cnidarians, bryozoans. It's the second most biodiverse habitats in the Mediterranean. And all of these three-dimensional organisms are also considered endangered species because they participate in creating and maintaining these habitats and its ecological function. So basically, the aim of my study was to try and monitor the best way possible the 3D complexity of these organisms. And for that, I was using the emergence of new technology, especially innovative non-invasive 3D approaches to try and obtain a better accuracy and repeatability of the monitoring of the 3D methods. Therefore, I was trying to assess the restriction success of two transplantation campaigns of two characteristic engineering species of collagenous, Paramoichia clavata and Covadonobum. So I was monitoring these two populations, and my goal was to try and optimize the monitoring protocols for this restoration action. And for that, I needed to assess the relative cost efficiency, the practicality, and the potential of these new methods compared to the traditional 2D photographic approach. First, a little bit about my area. It's the same one as Sarah just presented. So Gallinara Island that was defined as a site of community interest and a special conservation area. And in this island, you have 4 characteristic habitats of the Mediterranean, including the collagenous habitats. Specifically in Ponta Xiaosha, there was 2 restoration projects. So first, there was a restoration project of the Gorgonian Paramoichia on a collagenous terrace at 38 to 42 meters depth. And there was also the restoration of Corallodonobum population in the caves and overhangs at 32 to 35 meters. I will first start by presenting the restoration and the monitoring of the red coral population, and then we'll move on to the Gorgonian. So, just a little bit about this species. As maybe you know, it's been longly exploited for derogatory purposes, and that's why the populations in the Mediterranean have severely declined in the last decades, and it's not protected under the Annex 3 of the CITES Convention. It's a long-lived and slow-growing species, and its characteristic is that it hides in overhangs and caves, and that's important for the monitoring process. So, first of all, one most efficient transplantation technique was used through epoxy, putty, and grits to maintain the new transplants that were obtained from the Portofino MPA. This restoration action took place under the National Italian Plan for Resiliency and Recovery.
And it has been almost 8 years now that this restoration process has been taking place. And in total, we've had 62 transplanted colonies that have survived the transplantation process. And now I wanted to show you how the 2D traditional approach looks like. Because it's in caves, it has a lot of constraints for the diver to go and take close-up imagery. So here you have a little time scale of the same 2 colonies in cave. Because of the constraints, we're using the number of apices, the number of branches variation, as a proxy for growth. And the main results that we obtained were a survival rate of 67.7% after the 8 years of restoration. And we also computed like the total number of apexes in the restored colonies, which was shown to decrease slightly, but not significantly, sorry. And we also computed the main apex count per colony that you can see here for each year. And we started with 5.4 apexes per colony, and we went as down as 3.3 apexes per colony for 2025. And so the survival rate in the literature, because after a certain stabilization period, basically all the studies show a very similar range of survival rates. And for the total number of apexes decreasing, this can be partly explained by the fact that the donor colonies, so the healthy adult colonies, are still undergoing increasing pressures, so they're reducing in size and it's more difficult to get bigger transplants. The loss of apex count per colony was explained actually in a study by Ronetta and Ali in 2023, which is basically explained by an acclimatization process of the colony, so it's called autotomy of apices, and it's a response of the species to induce thermal stress. And now we're switching to the Gorgonian restoration projects. So again, just a little bit about the species. It's also a long-lived species that has a very deep range. And its fan orientation is also always against the current flow, which is a little important for the restoration process. And this species, even though it has experienced general decline in the Mediterranean, has no protection status so far. And this time, we were able to apply 3 monitoring methods to the transplanted colonies. In total, over 100 transplanted colonies were present on the coralligenous terrace, but we only monitored the 22 first individuals that were transplanted in 2023. So the whole study is based on a subset of the general population. I'm going to present very briefly the different methods, but please ask questions if you're interested in the details of the methodologies. So first, the classic traditional approach. As you can see here, it's the photo quadrats with a framing scale in the background, and we extracted morphometrics, so maximum height and width for each individual colony. And here I also wanted to show you the time scale, so how it looks like after a few, so 10 months and then 13 months for the same colony. Then I'm going to present you the structure from motion technique, which has been applied in terrestrial environments for decades and is now being developed in marine habitats more and more. And basically, the principle is that we use a set of overlapping pictures to reconstruct a 3D model. So the application is very simple. You have a diver with a camera that will basically capture the whole study area. And here you have just a scheme showing the material that you need. So also just a scaling frame. And we extracted the same morphometrics on the point cloud. So I'm sorry, I did something wrong. Okay, everything good. Sorry about that. So we extracted the same morphometrics, maximum height and width, from the point cloud, and additionally, we also obtained population density and information on the taxonomic cover of the community. Now, the third method has been developed originally for and it's the first time it's being applied to benthic habitats. So the concept is you have two cameras on a grid. You have a calibration frame that you can see here on the left of the screen to be able to correct lens distortion and do 3D scaling. And then from the videos, I hope it works, I just wanted to show you how the transects. And here are the first results. So this is just for the subset population and just with the 2D photography results. So here you can see basically the overall temporal change of the population, and we did so thanks to repeated measures ANOVA, a significant variation in height over time, with a significant increase between 2023 and 2025. But we saw no effect of time on the width variation of the studied colonies. And the growth rate we found was 1.23 centimeters per year, which is a bit lower than what in the literature, which can be explained maybe by the recent process of restoration. And now I wanted to discuss a little bit the comparison of methods that I did. So here you have a box plot showing the morphometrics extracted in March 2025 for all three methods together. And the pairwise comparison showed that there was no significant difference across all the width measurements. However, there was significant difference between the height extracted in the classic methods and the structure from motion methods, and almost a significant effect between the heights of the traditional methods and the surveyed methods. And this was consistent with the findings of the linear regression models that we performed that show that the width is very well explained with the two innovative methods compared to the traditional methods. So we have an R-square of 0.7 for structure from motion and 0.8 for stereo vision. However, the relationship is not that clear for the heights for both methods. So we still need to keep monitoring to increase the number of data points and be more sure of our conclusion. And here is just a little bonus, because we don't really have time to go into details. I just wanted to show you the potential of the structure from motion methods. Here you have the whole terrace from the top, it's called an orthomosaic, and here is a heat map of the population density. And here you can see the taxonomic analysis that I did. So you have actually taxonomic information and spatial information on the whole terrace. So to conclude, is there better methods? Yes and no. It really depends on what you're looking for and what you have. So the 2D photography method is still widely used, and it's still widely very cost efficient when you're looking at shallow habitats and simply structured organism. The processing time is nothing, and you obtain individual morphometrics. However, for all three-dimensional organisms, it has several limitations that we're trying to compensate with these new methods. So structure from motion is a very full of potential methods. However, as the deeper you go and the more you increase your diving time, and if you want a good resolution on your 3D models, you're also going to have a big processing time. So you can obtain individual morphometrics, but you can also obtain a lot of community composition information. So my advice would be to use these methods only once a year or once every 2 years to be able to assess the long-term spatial processes and changes inside a community. And now for the new stereo vision methods, new because it's the first time we visit invented habitats, especially in our case of deeper habitats, it's very interesting underwater because it's very cost efficient, and the processing time, once you know how to apply the method, is also very fast. And the software you use is open source, so very easy. And hopefully, with further monitoring campaign on the same individuals, we can obtain more accurate and more precise morphometrics. So it could gradually, hopefully, replace the traditional 2D photography methods. Thank you so much for your attention. I really enjoyed my thesis, so I hope you enjoyed my presentation. And if you have any questions.
very much, Noor. I really enjoyed your presentation. I appreciate a lot the introduction for the topic, particularly for people that are expert products and those species. And it's true that I was writing some questions, but you are also answering at the same time, so I really appreciate that also, because you think about what the jury can ask me. And every time I think, oh, she has the answer in the next slide. So thank you very much. And I have just a curiosity or a thing that I was might be in my mind. It is who took all these images? And you were diving, or they need to be researchers? It could be any divers from Citizen.
Well, it depends on the methods we're talking about, but honestly, all three methods, you could train basically anyone. Actually, in the zoology lab, they are training citizen science program to do structure for motion. So this is a method that can be applied by non-scientists. And in the same direction, stereo vision is also just two cameras on a frame to do video transect. So as long as the monitoring protocol is tuned and precise, I'm sure that non-scientists will also perform this. So this is also why all these methods have a lot of potential, according to me. The monitoring campaign has been going on for a while for the red corals since 2017. So there have been several divers who took all the pictures every time, went as much as like 3 or 4 sampling times a year. So we're a lot of different divers. For structure for motion, it was always the photogrammetry team from the lab, so my supervisor including. And stereo vision also, it's the first year we're applying it. So again, I mean, we learned the technique this year. So it's all new. That's why we need a lot more training for this. But hopefully, it will give interesting results.
Okay, thank you very much.
Thanks.
Okay, yes, thank you. So maybe I guess this one, I felt the same, like you already answered my question directly. So, okay. But about the chat about the increase we talked about the increase of engagement of the colonies.
Yes.
And maybe I missed the point. You said that this is about the method, or could be other external factors influencing
The growth of the it's just that we didn't find any like significant increase or decrease of width over time. Yeah. Yes, but I mean it's not significant, but yes, there's a lot of variation also because between the different colonies that are monitored, there's a huge variation in the range of the width. So like some colonies had 1 or 2 branches, so obviously the width is very small. I think as small as like 0.8 centimeters, and some of them were very large. So I think that also might explain the huge variation that prevent us from seeing like temporal effects. Yeah, also, again, the width for 2D photography is very hard to capture because, like the 3D complexity of the organism. If you have a crooked branch or if the organism was moving in a different direction at 2 different sampling times, then maybe when we're doing like the image J analysis, you can have very different results. So that's one of the big limitations of those methods.
more about what feeling is more about the methods than other courses.
Yeah, for me, because this result is only from the 2D photography results. So I think for me, I felt like this was more because of this. Yes.
Just like one slide when you are preparing. Yes.
So from the photos you needed columns, right?
Yes, with the 3 methods we use the same subsets.
Okay, so I use the particular thank you.
For the 2D model, you were only comparing with height. Would it also be an option to compare more like the distance from branching, or no, how is the current growing? Is it like growing like a tree from upwards and like growing from the bottom and like elevating everything.
So wait, maybe I can show you with the, oh my god, too much animation, sorry. So just here, I mean, so basically the colonies, once they're transplanted, because they're 3D, so it's a little hard to show you, but they have maybe one or two main axes, and then they will expand both in width and in height at the same time. So that's why we're trying to grasp like the canopy, like a tree. That's why we're trying to use these two different methods.
But are they growing from the tips onward, or it's like the bottom pushing upwards and pushing like the newest organisms? Are they in the tips or
from the tips?
Thank you very much. Thank you so much. Yeah. Yeah. And actually,
But I feel like the one is something yeah, because some of my brothers is ego and mine was yeah, it's really interesting. Yeah, yeah, I mean, so growth really makes sense. Yeah. I was gonna be able, but they didn't have any work because it's all online. It's not like so working there, but I'm still struggling. I went through a nice conference
She's actually on the bus. Yeah, the car there, I don't communication, but he knows presentation, so it's fine. But yeah, no families and no life. What's happening? Searching, yeah. Wait for me. Why you choose this? Yeah. Yeah, there's a lot. Yeah. Yeah, it's really weird because YouTube's now really expanded. So they're everywhere. And you can listen to that we already have in So that's kind of my main highlight of the presentation. I'm like, okay, we should think about it more. That's my way. With the microphone, the camera. Yeah, no matter what. Probably from there. Is it far there? Yes. Yeah. Yes. Yes. Everywhere. Yeah. Yeah. Even outside today, I think I was really happy right, but there was no real trees. Yesterday was better. It's weird. It's a new campus. Have any AC inside? Yeah. Yeah. No. Just make it. Yeah.
Okay, so thanks everybody for waiting. We were waiting a little bit again to catch up with the streaming. So thanks to all our speakers that are keeping really on time, which is great, but it means then we don't want people to be online and missing the presentations. So we just wait a few minutes. So now I'd like to hand over to Maria, who's going to talk about your project on for environmental engineering you're telling me. So I look forward to hearing this one. Over to you, Beth.
Hello everyone, thank you for being here, and it might seem to you a little bit out of what you all doing. I'm referring to my colleagues, but also to the judge. But I have a really simple question to you. Did you check the news today? Or maybe yesterday, or the day before? I feel that we all kind of keeping up with the news recently, with all the political situation, with the crisis, with all this competition for a lot of resources. So, and in general, in climate change, it's quite evidential that we do need transfer to renewable energy. It's not an option anymore. We do need to go for it. We will go for it anyway. It's coming in the future.
However, the way we plan it will actually impact us and will impact the nature. So here I'm really pleased to welcome you to my presentation on the analysis of offshore wind farm locations in the context of marine spatial planning in Asturias, it's in north of Spain. So I will do a quick introduction why it's important and what EU has as a goal in Europe in general renewable field. I will explain you my methodology, it will be step by step. It will be a little bit technical, but I will try to make it as simple as possible so everyone can follow up and keep up with my presentation. And I will show you the main results. It will be a lot of maps, so later on we can always go back to the slides, and if you have some questions about it, I will address it. And the main conclusion. So first of all, the European Union aiming to reach 300 gigawatts of the renewable energy installed both on land and offshore by 2050. This translates into almost 94% of the expand of the energy production, but also infrastructure. So we need to put more and more of the wind turbines itself offshore and on the land. And usually these areas overlapping with really sensitive ecosystems, and also with the fishing grounds, with the interest for the communities, the coastal communities. Therefore, what is really important is to implement the nature-based marine spatial planning when we plan something for renewables. So the aim of this research was to find a suitable offshore wind farm locations with minimum conflict between different stakeholders in Asturias that has a valuable ecology and also has a North Atlantic wind resource that's quite promising. So, 3 objectives. Just to make it simple, my main objective was to analyze the area, so to create the baseline of what do we have in Asturias and what do we need to protect, to find the suitable areas, so within this, where we can go to minimize the conflict, and the third one is to compare these areas and to find the best one in terms of the energy. Just a quick introduction to study area. We have the Asturias, so my research was covering all the area of Asturias and the part of Galicia and Cantabria on the east and west, it's the north of Spain. It has a key biodiversity sites like El Cachuchis Mountains. From the coal industries, it was a traditional industry there from economical perspective. They transitioned from this now, and they showing the interest in renewable energy in general. Mainly it's on land right now, but they're also thinking about offshore expansion. And we have some main challenge from technological point of view. It's near-continental shelf. We do not have much of the space where we can install it. We have quite dense fishing grounds that's impacting both economy and social aspects. And also we have quite visibility sensitive landscapes, so we have quite high cliffs, and we can see the renewable energy from the coastal line. That's sometimes creating a lot of conflicts. Methodology, I have 3 objectives, as I already mentioned, and 5 main steps. I will guide you through this step by step. First is the data preparation. So as I said, I was trying to find the baseline, we understand what is there, what is on this side. I identified 27 local specific variables and divided them in 4 main groups. Here I would like to highlight that in similar studies they usually focusing on only physical variables, so basically energy potential and technological feasibility, and that's it. On this level the usual analysis is stopping. However, on top of the physical one, just speaking quickly about the physical, this is a set of equations that was used to interpolate the data. I would not go into details, it's too technical, but we can also speak about it if you want. We have ecological data that's interesting, I guess, to everyone in this room. I found the 5 main biological groups. It was birds, mammals, fish, plankton, and benthic species. I was not checking them in specific. I was more trying to find all the possible biological groups that could be impacted by the renewable energy in general and include them in my analysis. We have economical data, and we have the social variables also being a part of this overall assessment. These identified conflict zones. This is also important because there is an MPA already established, marine protected area already established in Asturias. There is a military training zone that was automatically excluded from all the analysis from the potential for renewable energy because it's a direct conflict, so we cannot install the renewables in the same area, whereas MPA, for example, already being established. So that was already excluded from the first slide. Then we go into the methodology of multi-criteria assessment. I identified 3 main scenarios. First one is energy-based. typically used for all renewable development is considered only wind power, seabed, and bathymetry, so basically energy potential and the feasibility of the energy itself. Second one is ecologically based scenario. That is what we most feel about, most related to in this room, I feel, is to protect as maximum as possible the ecosystem, to exclude all sensitive zones as possible. So there was a biodiversity index developed, and on top of this, there was a hard exclusion zones from the Marine Special Plan of Spain, because the Marine Special Plan of Spain also identified the sensitive zones, and it was automatically excluded. But what I would like to highlight at this stage, the scope of this research was not to evaluate the impact on the ecosystem. The scope of this research was try to find the balance. So we know that we have ecosystem, but we know that we need to go for renewables, and we need to find something, some areas that will be balancing all of them, and on top of this I included social variables and economical variables. And this is the last scenario, it's a balanced one with Marine Special Plan implementation tools, and this what was mainly based for the suitability analysis. So how to do it? It was done in QGIS, and 2 different applications was used. It's similar approach. We are given the weight factor for all of the variables, but we're doing it in different workflow. We either do it cumulatively, like the pairwise comparison matrix, so all of them going simultaneously and being analyzed simultaneously using all the variables. Or another one is a special floor when we given the weighting factor for one group, we excluded all the variables one by one. And it's important because in the end we will see that the results are quite different. And the third objective is to find suitable zones. 4 suitable zones was identified, and later on they was compared based on the first data prepared for the variables, but also the wind turbine was, I identified the Vestas wind turbine. Again, it's a little bit technical, but this is impacting techno-economical assessment. So this was needed to actually realize, okay, how much energy it would give us if we go for renewable energy. Because one story is to install something offshore, but another story is actually make sense of what we're doing there. And sometimes it's in conflict, so I was trying to address it immediately. And moving to results and discussion. As I already mentioned, 4 zones was identified. First one, it's highlighted here on the map, so all zones will be highlighted while I will be speaking about them. First zone have the most energy. It has the most wind speed, it has the most energy, however, it's the lowest from technical feasibility because it's having the lowest number of maintenance windows. I know that I guess now I lost a lot of you because this is really technical. Explaining quickly, maintenance windows when we can actually take the boat and go to maintain our wind farm, because usually the wave height, I guess we all experience it when we take in a boat and we want to do some field work, there is a wave height too high, so we cannot go there. And this is what the maintenance window is speaking about. So there is not much of the maintenance window where we can go and install it. And one story is to install it 1 year, but then we have it running for 20 years. So within 20 years, we need to be sure we can go there and actually maintain it and make sure that it's still operating. That's why it was really important. From ecological perspective, we have quite high bird diversity in this area, and it's highlighted on the map. However, and moderated benthic and fish. So generally it was moderated, and it's also located quite far offshore, so from visibility perspective it's not visible, so it's positive for us because we would not see any wind installation there. Second zone, moving here, it's further offshore. It has the biggest distance from the port and the grid infrastructure. Again, a little bit technical. Why it's important? Because when we install something really far away, we need to create a lot of cables that will be connecting the turbine itself to the grid, to the storage where we will be storing all of this energy. So as long as these cables will be, it's more economical, it will be more expensive, but also it's impacting more of ecological habitats. So we would prefer to avoid to put it really far offshore. So technical feasibility is moderated. There is more possibility to go there, but it's still quite far. From ecological perspective, we have quite high impact on mammals and strong for plankton and birds. And also at some point, it's overlapping with the key ecological zones. However, from visibility perspective, it's also not visible, it's far. Moving to third one. Similar results, we have lower energy yield compared to the first two zones. However, it's still classified as a zone for green light, so we still can go there because there is an overall classification for renewable energy that's advising to go for this time speed and not for this one, and it's still passing the limit. It has also moderated feasibility. From ecological impact, also quite moderate. It has the lowest mammal diversity area. However, it's moderated values for other species, so I would say it's also fine from ecological perspective. And what the problem in this area is that it's overlapping with the fishing grounds. And for the Asturias, it's really important, the fishing grounds for local economy and also from a social perspective, because it's artisanal fisheries, it's more than 70% of artisanal fisheries in the area, so we would prefer to avoid this. Again, it's quite far, so visibility is not affected. The last one, the most interesting one. It's located the closest to ports, it's visible, and it has the lowest wind speed and therefore the energy yield. However, from technical perspective, it's the most feasible one. Because in all the other three zones, we was implementing the floating wind farms. So there is a pilot studies on it, so they operating, on the floating platforms, but they not yet developed to the stage that we can actually go into next year, for example. So from technological feasibility, this zone is the most technically feasible, so probably they would go for this zone. However, from ecological, it has a sensitive zone. It was identified as a sensitive zone in Marine Special Plan in Spain, and it's also only one zone that has a visibility impact compared to other four. So, just to sum it up, we clearly see the trade-off. We clearly see that there is a trade-off between ecology, economy, and technical feasibility. And this will really impact who will be taking the decision of installing the renewable energy there or not, because there is ecologists who would be saying, okay, we need to protect habitats, so please, as far you go, as better. But there's also people saying that, yeah, if we go far, we cannot actually install anything because it will not work or we cannot maintain it. So there's a clear trade-off between different situations. And also we have a conservational dilemma. Because in this analysis, I was assuming that the wind farm creating the reef effect for benthic communities and for fish, because it's creating more habitat. And it was done based on the sandy habitats. So one analysis was to identify different types of seabed. So for sandy habitat, it was given as a plus, a reef effect. However, this is actual dilemma. Should we protect what we already have there, or should we install something more, but we increase biodiversity index? And this is what we need to address as marine biologists, and this is what the stakeholders should understand from us in as simple words as possible. So my main conclusion, we have four areas. We have a floating wind farms that is still not yet developed. We have a different methodology that actually impacts on the final suitability score. So when this type of analysis is being performed, we need to understand what type of methodologies it's using, because in the end, we have a different result. But what's the most important, personally for me, and I guess for everyone in this audience, is that this is really important as decision-making tool. Because at this moment, what is going on, usually it's a wind farm developers, they come into municipality, they assess the technical feasibility, so again, just energy basically, and they proposing to install it, they installing something, and then ecologist arrives, and they're saying, okay, it's already there, but we need to do something because it's impacting this, this, and this, but the cumulative study is done after the installation. And from my experience, I went to Wind Europe conference in Copenhagen, visit this preparation of this research, and this is being spoken up right now, but it's not yet implemented. And it's not yet implemented even from European perspective. So that's why I wanted to highlight this as a decision-making tool that we as biologists, we need to bring it more up, and we need to speak more about the fact that we need to balance not only ecology and technical, but also include social part and also include economical impact, because yes, we installed something interesting from economical perspective, but in the same time we impact in the fishermen, for example, that are locals and that are important for this area. So with this, I would like to thank you for your attention. I would like to thank my family who's been watching me online, and my supervisor, Zabiedli. And yeah, welcome to answer your questions.
I go. First of all, Maria, congratulations, because it's not an easy topic and you explained it very well. I think that you don't lose so many people, although the complexity of the topic. And also thank you, because I'm from Asturias and I appreciate a lot of the study. And as you mentioned, this is a very, very hot topic in Asturias. And I also appreciate the analysis that you include the fishing grounds, because the main problem with this development of the mills is the fishermen. And they directly say they don't want it there, and they will do whatever is needed for them to find there. So I think it's a case zone. Take into account for choose one zone comparing to the other one. And I'm going to go to a little more technical question. Don't worry, not about equation, not numbers. But I'm wondering, where do you have the information for the ecological impact? For example, mammals. Where do you take it from, and how good or bad are those information?
Yes, so firstly about the fish, I didn't highlight it during the presentation, but it's indeed, it was a key of this research, because fish was represented in 4 different groups of variables. So there was total 27, 4 different groups, and it was represented as biological group, obviously, as economical importance, because it gives, and it was only for local economy, so I evaluated the different fishing grounds, and I assessed the specific weight factor for the most important one, because we do have this information from the municipality of Asturias. They did this research on the fishermen, and also assess the next one as socially important, because we do have the map where they fishing the most, and we also have the information on the length of the boat. And the length of the boat means if it's an industrial boat or it's an artisanal boat. So based on this, it was also included as socially variable, and I was trying to put them as a key element in at least 3 different groups. And when it's coming to the methodology itself, it's really important because it's balanced on 3 different levels. So it's really important that you highlighted it, because it's definitely a key. And answering your direct question about the, just a second, I'll come back. Here we go. So it was assessed using the Shannon Biodiversity Index, because it's a way to balance the amount of the species, so the number of the species and their distribution in the area. It was based on the OPIS dataset that was obtained in R. The data was cleaned, so I must make sure that there is all the only scientific names of the species, that they're not repeating themselves, that it's not false observations. It was for different temporal scale, because the OPIS dataset sometimes for some of the species have really short one for like 20 years, and for some it has at least 50 years of observations. Therefore, I decided that Shannon Biodiversity Index will be the best.
Okay. And apart from the Shannon Index, I need more raw data, like the list of species or something like that. Do you work directly with this? Or?
I analyzed in my manuscript what is the mainly occurring species in the area, but I didn't work with anything except the OPIS dataset. So for ecological, it was mainly OPIS dataset. But I was analyzing it from different perspectives, so what is the most occurring one? And for fish, for example, I compare it to the fishing grounds report from Asturias, so I make sure that it's actually making sense. And but yeah, all the information is coming from OPIS. And we also discussed it with my supervisors that there's different data sets for biodiversity and for species, but specifically for this area, OPIS has a little bit more information than other data sets. So that's why we decided to go for OPIS.
Okay, thank you very much, Maria.
Thank you.
I really enjoyed it.
I had the same question. That is, how did you assess the ecological impact of what you reported? But I guess you answered. And so, I'm fine. But on your vision for your presentation, you made this document which was published from the easy to understand. I just want your opinion, because at some point you mentioned the floating fund. Do you think that this structure will not used right now, but maybe something that will be in the future? Do you think that it will have also the effect
It's interesting because you can see on the slide, this is a fixed, okay, you don't see. So this is a fixed bottom, so we indeed have more structure to provide the reef effect. However, for the wind flow technology that's currently developed, it's just the upper part. And for this one, the reef effect was not estimated. So 3 areas was going for this one without the reef effect, but for the 4th one that's located, that's a fixed bottom technology, the reef effect was estimated because we have quite a lot of the structure. But what is interesting, again, this is a little bit of input from the conference that I attended. They actively now developing their nature inclusion design for the wind technology. So they already having it in mind on the development stage, not introducing something, not already doing something on existing one, but for future wind farm installations. And they including actually the reef effect that was presented before me, they including kind of the structures for the floating ones as well. So potentially it can impact the reef effect as well. And what's also interesting is that for the wind farm offshore is becoming automatically the protected area, at least from a trolling, because no one can enter the zone. So this will work both for bottom fix and for floating, if it's answering your question.
Yeah. Question. And as I was thinking how important this study is, and do you think or your supervisor to present this study to the local people or fishermen or management or something like that? Don't stay just only in the university.
Yes, we will develop a little bit more of the analysis for technological perspective because one of my supervisors is engineer, so we're going to include a little bit more on this information. So it will be interesting for people from municipality because if it's an ecological one, you know that they barely like reading them usually. So we will include more of the information on money perspective, but also we're planning to publish it and we would like to address it at municipality to really show, okay, there's some money coming from, but also this, and that's how we can actually balance it. Because the final results of this, the suitable zones that I showed you, already included the information about the ecosystem, so it's already with the minimum possible impact of the ecosystem. So if they would go for the zones, it's already, for our perspective of marine biologists, it's already kind of perfect. At least it's better than what before. So yeah, we're planning to publish it.
It's a little bit similar to that question, I think, or builds on it, but I was curious if you wanted to kind of say, or if you got to the stage of analysis. So you said you included fish as ecological, economical, and social variables in your model. Did they come out as more important under one of those categories, or is that something that you can tell from your analysis?
More important from an economic perspective?
Something about more interesting, or whether it was as ecological variable, economical variable, or social variable. Did one of these add more weight? Or?
I would say that definitely economical one, but for simple reason, not because of its importance, but because we can estimate it in money perspective.
Okay.
So when we do have the label, it's easier to communicate it to everyone, and it's easier even for us to understand, okay, how it's important. So yeah, I would say that this, but what is really missing, and there's really knowledge gap on the impact of the renewable energy on the ecosystem in general, on the cumulative assessment. So we still lacking a lot of data. So I would highlight the ecosystems, but I cannot because I don't have much of the proofs to highlight.
You mentioned that you weighted some of your variables. How do you decide on the weight? Because I feel like this is kind of a crucial point for the outcome.
Yeah, it's a great question. It's always subjective. There is no methodology developed for making it objective because you're doing it based on your expertise. I personally did it based on the literature review and based on all the different reports that was published on the impact on the mammals separately from the birds. So I was analyzing it step by step. But overall, in marine spatial planning, it's always subjective. Like, it's just subjective, and you're trying to involve as much experts as you can. But usually the stakeholder decides it themselves, and if they do not like fish, or if they do not like mammals, or if they do not care about it, they will just give it a lower scoring. But for this analysis, for the balanced approach, I give it equal weight factor. So it was from scale 0 to 1, and I give to each of the groups 0.25. So I make sure that they are balanced within the assessment. Any other questions?
Okay, well done again, Maria. Thanks very much.